The information in this video is provided for informational and educational purposes only. Hey everybody, welcome to another edition of the Morale Monologue. My name is Michael Morale. In today's video, I want to talk to you about a new SMN boosting molecule that's showing a lot of promise as an add-on therapy. You know, I've talked about Scholar Rock's treatment, SRK-015, now known as apitegromab, and that how this is also going to be a combination therapy with Spinraza if and when it's passed by the FDA. Now, we don't know when this is going to be passed. We're hoping by the end of this year or maybe sometime within the first quarter of next year, 2022. But there's another company that's doing some research about how to boost the SMN levels of the SMN2 gene. We know that the SMN2 gene only produces about 10 to 15% of the protein that our body needs. And of course, the more copies of the SMN2 gene that you have in your body, the better off you are. But this other boosting molecule that they're talking about could also be used in combination with the others, or at least that's their hopes. So the company that I work for, BioNews, is the parent company for SMA News Today. And SMA News Today put together a great article regarding this new molecule therapy and how much of a promise we could see as SMA patients. So the video that I put together is very technical, and I've only highlighted the key points in the article, but I have linked the article down in the description of my video. So go down and click on the article and read it for yourself if you want to read everything about it. Like I said, I just touch on the highlights. So watch the video, and then toward the end of the video, I give you my perspective a little bit, but I'll come back with my final perspectives, and I'll tell you what I think about this new promising therapy. Thank you. Targeting a specific region of an intermediate molecule generated from the SMN2 gene increased the levels of functional SMN, the protein lacking in spinal muscular atrophy, in cells derived from SMA patients, a study shows. In addition, combining this approach with a splicing modifier similar to Spinraza resulted in even greater SMN levels than treatment with the splicing modifier alone. These findings highlighted the therapeutic potential of targeting this region, called 5-foot untranslated region, especially when combined with currently approved splicing modifier therapies. Future studies are needed to better understand the underlying mechanisms of this new approach and to test it in a mouse model of SMA before being able to move to clinical trials, the researchers noted. SMA is caused by low to no levels of SMN, a protein essential for motor neuron and muscle health, due to mutations in the SMN1 gene, while a backup gene, SMN2, is capable of producing SMN. A slight difference in its DNA sequence results in an event called alternative splicing that limits the amount of functional SMN it produces to 10 to 15%. Alternative splicing allows for a single gene to give rise to many different proteins. Just like in a recipe, adding or removing certain key ingredients, in this case, sections of genetic information called exons, can change the results, the messenger RNA, known as mRNA, and the final protein. Messenger RNA is a molecule derived from DNA and used as a template for protein production. In SMN2, this event causes the loss of exon 7 from most of its messenger RNA molecules, resulting in a shorter, poorly functioning SMN protein. Both Spinraza and Evrysdi, known as risdiplam, two of the three therapies currently approved for SMA patients, work by preventing exon 7 deletion from SMN2 messenger RNA, correcting its splicing and increasing the production of a functional SMN protein. However, targeting splicing as a means of increasing SMN levels has a ceiling effect, determined by the abundance of SMN2 transcripts, messenger RNA molecules, in cells, the researchers wrote. As such, increasing the total pool of SMN2 messenger RNA and boosting the messenger RNA to protein process are two potential strategies to amplify the effects of splicing modifiers. Typically healthy SMA carriers have only one mutated SMN1 gene copy and can transmit the mutated gene to their children. A child needs to inherit two defective SMN1 copies, one from the mother and one from the father, to develop the disorder. Based on the different effects in patients and carriers, and given the fact that SMN is involved in the formation of the splicing machinery known as spliceosome, the team hypothesized that in states of SMN and spliceosome deficiency, an increase in SMN levels may alter its own splicing. We suspect that the increased levels of total SMN messenger RNA is a direct effect of the 5' UTR untranslated region ASO, whereas increased exon 7 inclusion is more likely due to SMN feedback, the researchers wrote. Moreover, the team found that treating patient-derived cells with a combination of ASO number 1 and a splicing modifier similar to Spinraza resulted in significantly higher SMN levels than those achieved when treating cells with the splicing modifier alone. However, the team speculated that the combination treatment would not be superior to ASO number 1 alone, since it already promotes splicing correction with a shift toward full-length messenger RNA. Our results add to the current understanding of SMN regulation and point toward a new therapeutic target for SMA, the researchers wrote. The team is now conducting further work to clarify the new ASO's mechanism of action, which may reveal that this type of approach can be used to restore the levels of proteins missing in other diseases, such as Duchenne muscular dystrophy. Of note, a patient application has been filed for the 5' UTR ASOs described in the study, and one of the researchers is a founder and shareholder of PepGen, a therapeutics company targeting severe neuromuscular diseases. In layman's terms, this means that researchers are finding different methods of boosting the SMN protein levels on top of, or in addition to, the increases that both Spinraza and Evrysdi are providing SMA patients. While it may be a while before it goes into clinical trial state, those of us with SMA should be excited that these additional add-on therapies could be included in our future. Okay, so I hope you enjoyed the video, and I hope you got some information out of it that you can use as well. Like I said, this new boosting molecule therapy is showing a lot of promise. Now, they haven't even gotten to the clinical trial stage yet, so this is probably going to take quite some time to get accomplished. But just think of it this way. If we can use SRK-015 or apitegromab as a combination therapy with Spinraza, and hopefully maybe even Evrysdi, then on top of that, have another boosting therapy that will increase the SMN levels of the SMN2 gene, we could be seeing a really bright future for those of us with SMA, and I hope you're as excited as I am. Hey, if you enjoyed this episode of the Morale Monologue, give me a thumbs up, and please subscribe to the SMA News Today YouTube channel as well. We would greatly appreciate it. Remember, click on the bell icon. That way you'll be notified of any new videos that we produce. We at SMA News Today hope all of you have had a fantastic day. Do me a favor this upcoming week. Do something for yourself that's going to make you a better person. Until next time, take care of yourself, and I'll see you in a couple of weeks. Bye-bye.